Thank you. My name is Bernard Siegel. I'm the founder and co-chair of the World Stem Cell Summit and executive director of the Genetics Policy Institute. I'm delighted to welcome you back to the session today, and we are very pleased to have our next speaker providing the industry keynote address. As you've been networking through the first day of the conference and into the second day, I'm sure you've gone down to the exhibit hall. You've seen many, many companies that have decided to exhibit here, part of the burgeoning, nascent regenerative medicine industry. Surely one of the pathfinding companies, if not the pathfinding companies in human embryonic stem cell research, is Geron Corporation. You know, we all here have a hope for stem cell research and stem cells as a way of discovering the root causes of disease, using cells as tools for drug discovery, and one day leading to cell therapies where we might not need drugs or surgery, but we can regrow damaged tissue and organs using cells. Geron Corporation has been present at the creation of this, and under the very able leadership of our keynote speaker, Dr. Thomas Okarma, they have moved into something that is so exciting, so new, and holds so much promise. They've actually engaged now in the first approved clinical trial using human embryonic stem cells. Dr. Okarma has served as Geron Corporation's president, chief executive officer, and member of the board of directors since July 1999. I first had the occasion to meet him when I was just emerging in my interest as a stem cell advocate way back in 2003 at the BIO meeting in Washington. I have had the privilege of speaking at many conferences, actually, with Dr. Okarma. Tom has always been engaging, always informative, so I asked the Assembly at the World Stem Cell Summit to welcome Dr. Tom Okarma.
Thank you, Bernie, and welcome to everyone. It's a real pleasure to be here. As Bernie mentioned, and as most of you know, Geron recently received permission from the FDA to begin the world's first human clinical trial in acute spinal cord injury of a product made from human embryonic stem cells. Well, with that accomplishment comes the burden of responsibility to do it right. And for us, doing it right means the right cell in the right place at the right time in the right patients safely. So what I'd like to do today is share with you the highlights of our 28,000-page IND that underscores the safety of this approach, and secondly, which gives some reason to be optimistic for clinical impact. So Geron, as Bernie also mentioned, was here at the beginning. We funded the 3 labs in the United States that successfully derived human pluripotent cells, and thereby we got the leg up on ways to turn the embryonic stem cell lines into a variety of differentiated cells for different therapeutic purposes. And while I'm going to spend all of our time today on the OPC1 product for spinal cord injury and mention some possible line extensions of that same cell, it's important that you know we are making terrific progress on the other cell types. So we are in large animal efficacy studies now with our cardiomyocyte product. We're in large animal efficacy studies in the UK with our chondrocyte project for arthritis. We have rodent proof of principle for our product for type 1 diabetes. So the lessons that we learned that I'm going to share with you today on OPC1 are really applicable to all of the cells that will follow. We hope our second IND will be cardiomyocytes for heart failure. And perhaps a year or so from now, I can come back and tell you that story. So for all of our programs, and this is very important for the commercialization potential of the platform, our products are scalably produced and distributed. We are not doing individual patient therapy here. We are borrowing from the time-tested true schema of monoclonal antibody production and recombinant DNA production to do multi-dose production lots. The product coming out the door is frozen, where it is then stored and shipped for off-the-shelf use. We've invested very heavily in process development technologies, including a relationship with Corning that has resulted now in the commercial availability of the Synthemax surface, which is a polystyrene surface that's sterile that incorporates a synthetic peptide that substitutes for feeder cells and matrigel in the expansion phase of embryonic stem cell work. We are also working with GE Healthcare using their wave bag system as the next scale-up step because the bead technology shown here, which also incorporates that synthetic peptide, is exactly what we need to scale up the production from planar surfaces to bioreactors, and that's in the works. So all of our products are manufactured according to the general scheme shown here. So we use master and working cell banks as the starting material to assure product uniformity. They then undergo the expansion phase, and the expansion of the undifferentiated cell is a process that's uniform for all of our different differentiated cells. Here's where the uniqueness takes place, where the recipes to create cardiomyocytes or islets or chondrocytes or glial cells differs from one another, and then they're all put through the same formulation for cryopreservation. And all of our products are manufactured in this schema. So when we begin to consider a disease application, we take the role of tissue engineers, and we jump into the pathophysiology of the disease we're trying to treat. And in the case of spinal cord injury, this cartoon tells you a bit about the problem that we hope these cells will serve. This is a cartoon of a human spinal cord injury, and the first point it makes is that these injuries are rarely transections. They are contusions or bruises that create first a zone of initial damage that then enlarges over time because of the inflammation that occurs as a result of the injury. There are intact axons that are myelinated, but they are rare. Most of the axons that are intact around the periphery of the central cyst are demyelinated. They've lost their insulation. And that is the prime reason for the transmission from head to lower body and vice versa stops at the area of the injury. So our initial idea was to learn how to make oligodendrocytes, the cells that produce myelin or insulation, inject them directly into the lesion site in the hope that they would remyelinate or re-insulate the axons and thereby restore function. And here it is. This is a frozen vial of OPC1, which is the product that is now entering human clinical trials. It is a well-characterized composition of progenitors to glial cells, and all of our products are always progenitors, and it'll become clear why we do that in a moment. What we have learned, and I'll spend some time sharing that with you, though, is that these cells do more than just myelinate. They secrete dozens of neurotrophic factors, and they also induce neovascularization in the tissue in which they are injected, which has huge implications for what these cells may be used for in addition to spinal cord injury. So the first thing we did in collaboration with the Kierstead lab was learn how to make OPC1 cells from embryonic stem cells. And this shows the sequential addition and removal of growth factors that at the laboratory research level resulted in the expansion of the ES cells and their differentiation into glial progenitors. Now, that was a very great step forward, but it's not sufficient for entering a human clinical trial environment. We had to take that academic process and industrialize it, create a system that's consistent with current good manufacturing processes. And that's what you see schematized here. So all of the materials that go into the process have to be highly characterized. And that's a bit of a challenge because many of these reagents do not come with a certificate of analysis. We had to invent and discover appropriate criteria to accept or reject a particular lot of starting material. So we generated the specs for all of the reagents. We have to worry about adventitious agents. So if a particular reagent had come into contact with horse serum, we are obliged to screen the final product for viruses that could come from horses. Then there is the operation, which has to be codified. So the unit operations, step A through Z, are all written down with very specified steps at which time quality control assays are interposed, and their results are permanently recorded. So the batch record for every production lot is a permanent record that enables us to certify or reject the utility of that manufactured lot. And then at the end of the day, it goes into cryopreservation. Now, the biggest difficulty here is characterizing the cellular composition. Because these are progenitor cells, they don't come out the door with little signs on their surface telling you that they're the right or the wrong cell. They're progenitors. They are obligatorily destined after injection to turn into oligodendrocytes, but because they're progenitors, we had to invent the appropriate release specs. That required us to do actually a very deep dive into the genomics, the DNA of this cell population. So we screened them with microarrays, and then the signals that we got that we thought were important were then amplified by RT-PCR. And in the release specifications for the product, we use flow cytometry, we also use RT-PCR, and immunocytochemistry, not only for the purpose of assuring that the product is appropriate, but also to do retrospective analysis correlating the release specifications with the behavior of these cells in various efficacy and toxicity studies, so that the correlation between the characteristics of the cell and their behavior in an animal model was absolutely paramount. So many of our assays, as I mentioned, are flow cytometrics, sort of shown here, both assaying for the right population of cells and assuring ourselves that the population does not contain cells that we are not interested in. In addition to the process, we have to have a facility that is appropriate for GMP. We have in California the only state-licensed facility to manufacture embryonic stem cell-based products for human clinical use that we think exists in the world. So there are 2 Class 10,000 clean rooms. There are protocols for how one gowns before you enter the room. There are maintenance requirements that are applied periodically. And the room and the people in it are quality controlled as well as the process. So once you're done with all of that, then you can start delving into the IND-enabling studies, because you have to demonstrate with cells that you make according to the way you're going to use them in the clinic that these cells are safe and effective in animal models. So the first thing we did was validate that these cells myelinate. And one of the animal models we used is the shiverer mouse, which is genetically incapable of making myelin. And so a cross-section of the shiverer mouse's spinal cord is here, and you can see the absence of multi-layered myelin around the axons. A single injection of the human cells results in, under high power, compact human myelin, providing good evidence that these cells do in fact mature in vivo into myelinating oligodendrocytes. We can also label them with green fluorescent protein, and in the right panel you can see the physiology of these cells in our spinal cord injury model. So if you follow a particular labeled cell, you can see that it myelinates multiple axons in its vicinity, which is precisely what your glial cells and mine do in our cords today. So for all of the myelinating properties, these cells manufactured in the GMP facility absolutely replicate the myelinating physiology of human glial cells. But they do more than this, and this is really where the excitement for future applications of the cells comes from. They make dozens of neurotrophins, proteins that enable nerve cells to withstand stress and to do sprouting. So we can assay this in terms of a potency assay by taking the media in which we are producing the cells and exposing them to a rat sensory neuron assay. And in 24 hours, you can quantify the neural sprouting that occurs because of the family of neurotrophins that are secreted by OPC1 into the media. And this is a convenient potency assay for the FDA. And lastly, these cells create new blood vessels. And this is shown in animals in a dose-response way. So here in brown are the human nuclei from OPC1 in the animal, surrounded by multiple blood vessels that are of host origin. And this is dose response, that at the higher dose tested in the spinal cord injured rat, nearly all of the animals develop new blood vessels. All of this is good for spinal cord injury, but as you'll see here at the end, it also has implications for other central nervous system diseases. So in short, we've discovered that these cells are the nursemaid of the central nervous system, and that never could have been discovered had we not been able to manufacture at scale and at high purity these cells, because you can't get them out of an animal or out of a human being. So the system in which we test safety and efficacy is shown here. Under anesthesia, a rodent has its spinal cord opened, and then a device called an impactor delivers a very reproducible blow to the spinal cord. And we do that in the thoracic region bilaterally, and we can do it in the cervical region, either unilaterally or bilaterally. The cells or the control vehicle is then injected within 14 days of the injury, and we then follow the animal for a variety of endpoints. So here is an example of what these cells do and what the injury does to the animal if untreated. So if you could animate the animal on the left, this is a control animal that received the injury, but in this case just got fibroblasts. So the animal walks by dragging its tail on the cage floor. You can see its hind paw placements are abnormal. The animals are incontinent, and they have truncal muscle weakness. They are unable to stand on their hind limbs. In contrast, here's an animal on the right that received a single dose of our human cells. And while the animal is not normal, you can see the tail is off the cage floor, the hind limb placement is more normal, and there is some return of truncal muscles, although the animals are still incontinent. Now, this can be quantified in a number of ways. The so-called BBB score is shown on the upper right. This is the level of improvement that is occurring in the control animals. Animals receiving various preps of the cells are significantly improved permanently. We can also do kinematics. We can take movies of the animal ambulating in its cage, and that's shown here in the lower left, where we can see that the uninjured control is almost indistinguishable from the animal receiving the embryonic stem cell prep with regard to stride length, stride width, paw rotation, and toe spread. So there is a significant and durable improvement in the animals that receive a single injection of cells. Why is that? Well, remember the cartoon that I showed you at the beginning of the human pathology. On the left is what you actually see in the injured animal 9 months after the injury without treatment. So there is your central cyst that's large, and there are some myelinated axons in the periphery, but if you look at high power on the edge of the cyst and stain for myelin in blue, you don't see any because the axons are demyelinated. So it's a very good model for the human condition. In contrast, on the right panel is an animal 9 months after injury and injection of a single dose of the human cells. And look at the difference. The cyst is filled with human cells, stained in brown, that are myelinating axons that traverse the cyst. So this is what regenerative medicine promises. A single intervention that doesn't treat a symptom, but it permanently restores structure and function of the tissue that has been damaged by a disease or injury. We show the same kind of data in cervical injuries. Here's the control months after the injury, where there's enormous loss of tissue. In contrast, animals that receive in the cervical region a single injection of the human cells have massive sparing, not only of the white matter, the axons, but also of the gray matter, which is the cell bodies. That's not due to myelination, that's due to the secretion of these neurotrophins that we talked about. Similarly, in the cervical model, we can demonstrate that this regeneration in the tissue that we show is correlated with improvements in forelimb locomotion that are statistically significant and durable. Now let's jump into the spinal cord and get a sense of the mechanism of action of these cells and the progression of differentiation from progenitor on day 1 to myelinating oligodendrocytes after several weeks. So in the beginning, on the top, this is 6 months after injection, you can still see some progenitors. There's very little mitotic activity, so 6 months after the injection, all of the numerical amplification that I'll show you in a moment is largely finished. And you don't see many neurons or astrocytes, but what you do see are explosions of normal human oligodendrocytes. This is a dual immunocytochemistry stain that looks for human cells plus a specific marker of the kind of cell we're looking for. So this is after 6 months. Compare that on the bottom to an animal after 6 or 7 weeks. This is the border of the rat tissue, and this is the border of the human tissue, stained in green. And there aren't too many, there are a few, but there aren't too many mature oligodendrocytes after 6 weeks. So this is a progressive differentiation that occurs after the cells are injected. Now, a very important question that the FDA rightly asked us is, well, where did these cells go? So this is an experiment where we learned exactly that. In order to do this work, though, we had to invent a new assay. We use a PCR assay that detects a human repeat sequence called ALLU, and it has a limit of detection of 100 femtograms of human DNA. So what this graph shows is the progressive migration of the cells from the point of injection. So here we're looking at a low or a high dose 2 days after injection, low, high dose 2 weeks after injection, not much difference. But after 6 months, you can see that the cells migrate a total of 16 millimeters north and south from the point of injection. We do know that the cells remain within the lesion site, as you'll see in a moment. They do not go beyond it, but they do fill the lesion space, which is an important consideration for the design of the trial. Now, this is a really important slide. It makes 2 very important points. First, that the cells remain in the thoracic cord. So what we're looking at now is the quantification of human DNA, not just the detection, but the quantification. So here on the left group, you see the low dose of cells given in the thoracic cord after 2 days, 14 days, and 6 months. And you can see a progression of the amount of DNA. That's because the cells are amplifying numerically. They are mitotic. They divide. So you inject one cell, and months later, you actually get 6 to 10. You can see a dose-response curve here. Here's the high dose given in thoracics, 2 days, 14 days, 6 months. And here, 6 months of the higher dose has many more cells than 6 months after the lower dose, proof of the amplification of the cells after injection. That's the first point on the slide. The second is that the cells remain in the thoracic cord. So if you look in the cervical cord at a high or low dose given in the thoracic cord, you don't see any counts. If you look in the cerebellum, the base of the brain, after a thoracic injection, you don't see any counts. So the cells remain where they are injected, a very important principle. Now, the T word, teratomas. This is a huge issue. Most academics who don't go through the labors that I shared with you to make cells under GMP have contaminating live, undifferentiated stem cells in their preps. That's to be expected. And so there's this notion that one of the problems with embryonic stem cell therapy is the obligatory formation of a teratoma. Well, we studied this rigorously. The entire spinal cord, brain, and body of these animals was examined by a veterinary pathologist looking for teratomas. These animals were kept alive for 12 months after the injection. We found 0 teratomas out of over 400 animals injured and injected with the cells. So the teratoma notion, at least for this cell type, has really been put to rest. What we did find are these microscopic epithelial cysts, which in the beginning were present at an acceptable frequency, but we had a bad study occur in the cervical region shortly after we were given permission the first time to start the trial. That was last August. So we were put on hold again because that animal study questioned the precision of our release specs. So we did the deep genomic dive that I shared with you and had to do another series of prospective studies to show that the new specs accurately predicted a cystless animal. And that's the data shown down here, where we have one of these things out of over 100 animals followed for 9 months. So we are pretty confident in the absence of any kind of ectopic tissue. And in none of the animals who got these cysts was there any evidence that it was harmful. They were microscopic, they remained in the cyst cavity, they didn't go anywhere, and there was no evidence of any impact on the animal. So then there's a whole list of more standard toxicology studies that I won't bore you with in terms of the detail, but you go through a lot of different parameters to demonstrate in a more standard way that the injection of these cells does not interfere with the organ systems of the animal. Particular attention was paid to allodynia, which is the artificial sensation of pain that many spinal cord injured patients suffer from. And it was important that we showed the injection of these cells does not increase that phenomenon. So we actually tested that in hundreds of animals for both mechanical and cold and heat stimulation and found no difference between the controls and the animals that received the cells with regard to allodynia. Now, the immune story is really very interesting. You also hear in the literature that this is an allogeneic cell. It's made from a particular stem cell line. Therefore, it's like a kidney transplant, and we're going to have immune rejection issues. Well, we actually don't find that in vitro. So both the undifferentiated embryonic stem cell, OPC1, this cell type, and our cardiomyocytes are not recognized in vitro by the human immune system. Neither sera nor NK cells nor T cells from a panel of unrelated haplotypes recognize these cells in vitro. What's that about? Well, consider the immunology of implantation. From the perspective of the pregnant mother, the implanting blastocyst is like an allograft. It's part mother antigens, part father antigens. But the mother's immune system doesn't reject immunologically that implanting blastomere because nature has figured out how to prevent that from happening. And there are characteristics on the surface of the blastomere cells that prevent the maternal T cells from recognizing or activating from the implanting blastomeres. Those traits are inherited by the undifferentiated embryonic stem cell, which is derived from those cells, and it's passed on, at least an early differentiation, to cardiomyocytes and OPC1 cells. So this is the basis for, as you'll see in a moment, our low-dose and temporary immune suppression in the spinal cord trial. So the summary of this is we did 26 protocol studies involving over 2,000 spinal cord injured rats, over half of whom were injected with the GMP-produced product, requiring 6 times 10 to the 9th OPC1 cells to be produced for these studies under GMP. And the list of findings, that most of which I've discussed with you, appears on the right, so I won't repeat those here. Now, we weren't done after that. We needed to provide the neurosurgeon with a reliable delivery mechanism, so we invented and qualified a syringe positioning device, which is a device that is transported to the operating room in a SteriPack. Part of the training for all the trial sites is that the surgeon needs to assemble the device aseptically within 15 to 20 minutes. And you can see here that it hooks onto the frame of the operating room and provides a 3-axis drive to enable the neurosurgeon to precisely place the needle tip in the area of injection and to carefully control the depression of the plunger. Once that package was put together, then we had to engage a variety of people and committees to devise the clinical protocol, which had patient safety as its prime and foremost objective. So all of these bodies, some at an institution, some at the agency, some that had jurisdiction over multiple institutions, were engaged sequentially to be sure that the way in which we were designing the trial maximized patient safety and gave an appropriate risk-benefit equipoise. So all of those conversations resulted in the clinical protocol that's schematized here. It's open label. It's subacute, functionally complete T3 to T10 lesions. So that's the thoracic region. Why did we start there? Because you may know that thoracic injuries are rarer than cervical injuries because of car airbags. The reason, again, is patient safety. So if I'm a T3 paraplegic and I get the injection of cells, if there's toxicity, it has to be manifest toward my head, T2 or T1. If I have that progression, while not desired, it doesn't significantly impact my long-term care. Consider, however, if we had started with the cervical patients and were I a C3 quadriplegic and got injections of the cells in that region and had toxicity and became a C2 or a C1 patient, I could lose respiratory drive. So even though these are rarer patients with a lot more comorbidity, we've agreed appropriately to do the first safety study there. We will have temporary immune suppression with very low doses of tacrolimus. I'll show you that in a moment. Clearly, the primary endpoints are safety, both neurological and overall, and there are a series of validated physical exam algorithms or surveys that will be applied in a reproducible manner for all of these patients over a 5 to 15-year period. In terms of imaging, there are 9 full CNS MRIs done in the very first year, not counting the validation, inclusion-exclusion MRI that occurs before we ask the patient for informed consent. And this is a partial list of the instruments or physical exam algorithms, all of which have been validated, and we have to train all of the physiatrists and the nurse practitioners, because after the patient is discharged from the rehab center to home, since we're following these patients for a very long time, these providers have to get in their car and drive up to 500 miles to the patient's home, where these instruments will be, again, repeatedly administered. So this is the time schema. So the injury occurs on day minus 14. All of the inclusion-exclusion MRI stuff occurs here. Informed consent is obtained. And then day 0 is when the patient is injected. This is usually after spinal stabilization surgery, so it requires a second trip to the OR. It's also when we begin low-dose tacrolimus, which is tapered starting on day 45 to 0 at day 60. There are actually several protocols rolled into one: the intensive protocol for year 1, a slightly less intensive protocol for years 2 through 5, and then a yearly follow-up for 15 years after injection. So this is one copy of the IND, 22,500 pages, doesn't count the additional information that occurred since last August to get the trial started. It's the largest IND that the FDA has ever received. And it does speak to the unsustainability of this whole process. Whether you're a wannabe company like a Geron without a lot of significant revenues or a Merck or a Pfizer, a $45 million exercise to enable a safety study of up to 10 patients is very difficult to reproduce. So we hope that we get better and the agency gets better and gets more comfortable with this approach so that our cardiomyocyte IND, our line extension INDs for OPC1 are easier and less expensive to achieve, still without compromising job number 1, which is patient safety. So my last slide is just a forward look now. So we're now recruiting screening patients in 2 U.S. medical centers. By year end, we expect to have 4 to 6 up in the spinal cord trial. But we now know, as I've shared with you, that these cells, in addition to myelinating, secrete dozens of neurotrophic factors and provide neovascularization. So they spare tissue and they allow tissue to be repaired in the nervous system. That opens up some very exciting line extension possibilities in addition to spinal cord, including stroke, Alzheimer's, leukodystrophies, and multiple sclerosis. And we are now engaged with NIH-funded collaborators looking at animal models of these diseases to try to create a rationale for extending the current IND into some of these devastating disorders. So what you've heard here is chapter 1 of what we hope to be a very exciting story of regenerative medicine that segues from the daily consumption of pills that treat symptoms to a single intervention of live cells that permanently restores the structure and function of tissues that are damaged by disease or injury. It's a high-value paradigm that we think deserves the attention and support of the U.S. federal government. So if you agree with that, please write to your congressmen and women and get them to understand the significance of the Lamberth decision on this kind of work. It doesn't just affect Geron and our NIH-funded collaborators. It affects the whole field of cell therapy, because whether you're working with embryonic stem cells, iPS cells, adult stem cells, most of those NIH-funded grants have a control arm of human embryonic stem cells, because it's still the gold standard in vitro against which the results of iPS or adult cells are compared. All of those grants are threatened by this Lamberth decision. So it isn't just the voice of industry. It's the voice of groups like yourselves, patient care advocates, academics, friends of the field, or people who hope that when pills and scalpels are no longer effective for your disease or for the disease of people that you care about, that there's something more potent than just putting bone marrow into your heart after a heart attack. The job is yours as well. Thanks very much, and I'll be happy to answer some questions. I am blinded by the lights here, so you'll have to jump up and down in order for me to recognize you.
Question. The spinal muscular atrophy, you are not going to take a Geron OPC1 to that effort?
Could you repeat the question? I had a little trouble hearing it.
Spinal muscular atrophy. Is there a connection there?
We are not currently exploring that alternative, but it's a fair one to ask about. I think there's a question in the middle there.
Right here. Dr. Okarma, I've been paralyzed for 25 years, and I just want to say on behalf of many of us who are here today with spinal cord injuries, thank you so much for what you're doing to make our lives better. We really, truly appreciate it. My question is what do you think? I mean, one of my big issues is that I think for all of us to come together, and you just said it in your final remarks, is for all of us to come together and put pressure on our legislators in Washington. And it would be helpful with industry leaders like yourself if we could get to Washington as patient advocates and scientists and industry leaders. And I just wanted to know if you would be in favor of getting involved in that effort.
Well, that's very well said, and thank you for the compliment. I should also say that all of us owe a great deal of debt to the 8 to 10 spinal cord injured patients who are going to sign up for this trial. They are truly the heroes of this effort. With regard to your question about patient groups and patients advocating for this therapy, we absolutely agree with that. This morning we had an Alliance for Regenerative Medicine board meeting where we spoke exactly to that point. We had a fly-in a few months ago where about 30 people that are industry and academia representatives went to Washington to meet with over 60 congressmen and women to point out the issues of the Lamberth decision and to advocate generally for regenerative medicine. And what was clearly absent from that effort were people like you or patient care advocacy groups that represent people like you. And so we've made a policy decision today to reach out to patient advocacy groups for membership in ARM and for inclusion in the next fly-in. So you're right on. Thank you very much. Thank you.
Thank you, Dr. Okarma. That was wonderful. And whenever they ask, what has human embryonic stem cells, what have human embryonic stem cells cured, you know the answer is that we've just barely begun. And when you look at the thousands and thousands of pages that has to be presented to the regulators, you can see that the road to cell therapy